Hello everyone, I am Omar Adrian Alaman, a new faculty member of the Biology and Agriculture Unit of PSHS SRC. Today we are going to review about key concepts that we need to know about molecular cloning. This video recorded lesson is intended to help the graduating cohort of grade 12 students in preparing for the UP College admission test as well as for the other entrance examinations that you may opt to take for your higher studies. In my slides for this review, I will be using pictures and guide questions to help you understand more on one of the many important techniques in studying molecular biology, that is, molecular cloning. Just like what you see on the first slide, we can compare molecular cloning to a photocopying machine. So we put a piece of paper in the machine with the intent to make many copies of it. And this is basically how molecular cloning works. In a nutshell, molecular cloning is the isolation and amplification of a specific DNA sequence and has 5 fundamental steps. First, we need to assess the DNA of interest that we want to amplify. Next is the use of restriction enzyme, or in some textbooks you might find it as restriction endonucleases. And then the use of a vector, or simply a vehicle where we can incorporate our DNA. And then after that, we put the vector with our DNA to a host organism and then be able to select positive transformants or clones. Here we can see a simplified overview of the steps that I mentioned earlier. And number one, we need to have a starting DNA material. So usually obtained via PCR, or it can also be a plasmid DNA from an allochthonous or external source. Then we need to incorporate restriction enzyme sites in our DNA of interest. Usually it's on the both ends, or the regions that will be flanking with the sequence of our plasmid where we intend to put it. After we do that, we have to digest our vector. So if the vector is circle, then we need to incorporate restriction enzymes in the vector that will cut at specific sites so that we can combine or ligate our DNA of interest to our vehicle, in this case, this vector. After that, we can proceed to transformation, which is basically allowing a recombinant plasmid. When we say recombinant plasmid, it's the plasmid that we use plus our DNA of interest to be integrated to our host cell. So usually we use E. coli. Okay? And lastly, we will culture the E. coli containing our recombinant plasmid DNA and then make many copies of it. So let's try to elaborate more about each step by using some pictures and schematic representations. For the source of DNA for molecular cloning, usually we get like a normal DNA. But in some cases, it would depend on the nature of your experiment or your study. We can actually get a complementary DNA, which is designed from an mRNA template. So basically, if you're going to use a complementary DNA, your intention is to study about the coding regions of the genome, because as you recall, mRNA is the only segment of the DNA that gets translated into a functional protein. Okay? Or you can also introduce intentional mutation. Usually this is done if you want to study how a gene works via loss of function. So for example, if the original sequence of your protein works, and then when you incorporated a point mutation, it does not work. So that means your first result is correct. So it's more of a validation type of experiment. Other sources of DNA that's used for molecular cloning can be from environmental DNA. That's for metagenomics, like it can be from water in this case, can be from the terrain, or even from the gut, which is a relatively new field for molecular biology. Next is, now that we have the DNA of interest, the next is to select a specific DNA sequences. Now take note, if we get an RNA or DNA from a tissue sample, there's a lot of DNA there. There's a lot of DNA fragments. Now, how do we select our target DNA? You might recall the basics of polymerase chain reaction, wherein we can amplify a specific segment of DNA. It can be a gene, which means it's an inheritable property of the DNA, or it can be just any other segment of the DNA. Maybe you want to study about regulatory sequences or transposons or enhancers for that matter. So if this is the whole DNA that you get, you can, and if this is your gene of interest, you can design synthetic sequences we call as primers so that we can make many copies of this. So if we try to look at the picture in here, this is this segment of DNA, and for us to make many copies of this, we are going to design these black arrows that we call primers. So primers would anneal at specific parts of the DNA, and if these primers can recognize this sequence, then we can make many copies of this via polymerase chain reaction. So if you want to know more about polymerase chain reaction, I'm going to make another review for it, another review lesson, so we can be on the same page. And then how do we know if we have successfully amplified our target? So this is by doing gel electrophoresis, wherein we run our PCR product, or what we also call amplicons, in an agarose gel. So an electricity will be run in the gel from the negative to the positive side. And then we know that DNA in nature is negatively charged because of its phosphate backbone, no? The PO3 negative. So that means that if we put current from top to bottom, then the tendency of the DNA will move from top to bottom as well, or that is from negative to the right. It will go towards the positive end. Okay, so that's why it's shown here in these bands. Usually for electrophoresis, the first well in our gel is the molecular marker, or simply these are specified lengths in terms of base pair of the amplicons. So usually, for example, this is 750 base pairs. So if your target showed somewhere here, then you can estimate your gene product. And if you can estimate your gene product, then that means you can also predict if that is your target sequence or not, given that you know how many base pairs are there in your target. Next, so the next is, how do we insert our target DNA to make many copies of it? So the best way to do this is the use of a restriction enzyme. Restriction enzyme was discovered by Stewart Lin and Werner Arber, wherein these enzymes got their name from the fact that they prevent invasion of foreign DNA, especially from bacteriophage. So bacteriophage are viruses that infect bacteria. So because bacteria, unlike higher forms of organism, lack an immune system, their mechanism to fend off invaders like bacteriophage and other viruses will be to cut specific portions of the viral genome. Because virus, when it infects the bacteria, for it to make many copies of the virus DNA, of the virus, it will inject its genetic material, and then it will hijack the the cellular processes of its host, and then when there's too much of the virus, it will disrupt, right? So this is what restriction enzyme also will be used for. This mechanism of bacteria was used by scientists in order to cut at specific portions of the DNA. So if we know which specific portion to cut from the DNA stretch, then that means to say we can also control where to put it. Okay, I hope you understand what I'm saying. So these are some of the classical examples of restriction endonucleases. Endonucleases. EcoR1, so as you can see, GAATTC, CTTAAG, which is its complementary, this EcoR1 will cut like a blunt-ended pattern in a blunt-ended pattern, which means hindi siya pantay. It's not the same. Okay? So, and you can see it forms like a palindrome. So, yung cut sa taas, sa forward strand will be the same cut to the reverse strand. It's just that complementary yung sa baba. Di ba here, it's G cut AATTC. It's the same with the reverse strand. G cut AATTC2. Yun yung um, sinasabi about restriction enzymes. So this is very interesting. The scientist who discovered this actually won a Nobel Prize. I'm not sure exactly when. So this is used for molecular cloning because it allows scientists to incorporate a foreign DNA that is not related to a plasmid or yung vehicle that we are talking about. Okay? So what we do here is in the blue, this is your DNA of interest, and then you are going to deliberately put these red ones here, or the restriction enzyme sites, so that your resulting amplicon or your resulting PCR product would contain these red sites. Now, what's the benefit of putting these red sites? Now, if we have this plasmid, as we said, plasmid, this is a circular DNA. If we try to recall again, this is part of the bacterial genome. So the DNA of the bacteria, they have two. They have their nuclear genome found in the nucleus, and then they have their separate plasmid DNA. So these plasmid DNAs are what we are using for molecular cloning. Okay, because it's easier to map out because of its small genome size. So for us to insert our DNA of interest with restriction enzyme sites, we also need to digest. We need to cut this plasmid DNA in a way that we can tailor fit it to incorporate our DNA of interest. So for example, I'm going to cut here and cut here. Then for me to cut here and cut here, it should have the restriction enzyme sites. Like for example, EcoR1 and PST1. So for me to insert in this site, I also need to add EcoR1 and PST1 in my gene of interest, so that when you introduce your amplicon, it will readily attach. Okay, I hope you follow at this point. So when they attach, the next thing that we are going to incorporate is what you call DNA ligase. Ligase means to combine. It's like a glue, kumbaga. Okay, so this is how we cleave our DNA of interest to our plasmid. Okay, and some of the notable features of a plasmid includes its cloning site, the one that I have mentioned. Okay, where we can put our DNA of interest. The restriction enzyme sites, as noted by the pink arrows, that means at this portion of the genome, this can be cut by this enzyme. At this portion of this genome, this can be cut by this enzyme, and so on. Okay? Origin of replication, as the name suggests, this is where the replication starts. Okay? It can be this way or this way. Okay? The orientation will not matter for the plasmid as long as we satisfy the ends of the restriction enzyme. Because if hindi nagmamatch yung end ni restriction enzyme at saka nung ating dinesign na DNA of interest with the target sequences, then walang mangyayari. Hindi siya ma-attach, kumbaga. Para siyang Lego that is very specific to one portion. Okay? So this is an example of a plasmid that contains, for example, an insertion of a gene like 12S, for example, or the COX-1 gene. Okay, and these right here are the restriction enzyme sites. So after that we have incorporated our DNA of interest to the plasmid now becomes a recombinant plasmid. Okay? The next step is to make many copies of the recombinant plasmid. Okay? Now, you might ask at this point, why don't we just do PCR all over again? Like, if we know our target and we want to study it, why don't we just make many copies via PCR? Now, that is actually true, but that would be very laborious. Imagine doing PCR all over again and again and again. Again, you're going to design, you're go you're going to design primers, you're going to run the thermal cycler. So it would be very laborious. And also, the advantage of using an expression system, in this case a bacteria expression system, is that you don't need to do PCR again. What you need to do is you can store the bacteria that harbors or that contains the plasmid of your interest, and then you can culture it again and then extract the plasmid, which you can later use for downstream applications. So that's why this is a very powerful tool if we want to study molecular biology. Now we have two types or two ways to incorporate the plasmid that we designed to our host. So you can see here the host is usually a bacteria E. coli. Okay, so for us to introduce this plasmid back to its host, we can either do chemical transformation in the form of heat shock. So we simply expose the cells, the competent cells as we term it, in a high temperature so that the pores of the cell will expand, and then via the principle of osmosis from higher to lower concentration, the plasmid would flow through, going to the inside of the cell. Or it can also be by electroporation or by the use of an electric shock. So the same principle also, the aim is to make the pores of the bacterial cell wall more permeable. Okay? Now, after we do that, of course, we need to check whether we are successful or not. So the next step and the last step is how do we check for positive transformants or positive clones? Okay? So how do we do that? There are actually two ways. Okay? Which are applied at the same time. So if we try to look at our previous example of a typical plasmid, we can notice parts of the plasmid genome that connotes or that commands for ampicillin or tetracycline resistance or any antibiotic resistance. What this means is if there was a successful insert of this plasmid, that means the bacteria will now have ampicillin resistance. So what happens is if this is your agar and you put ampicillin here, those bacteria which were able to harness the plasmid that you introduced will survive. Do you follow? I hope you do follow. Okay, tatagalugin ko. Yung bacteria na successfully na-induce natin yung pag-incorporate ni plasmid sa kanya will be able to survive if you put ampicillin here. Okay? However, kung hindi naman siya naging successful, ibig sabihin, it will not grow there. Okay? So, ito yung first step natin to check for positive transformants. The use of antibiotic resistance. Okay? Meron pa siyang second step. The second step is what we call blue-white screening, which is a rapid and efficient technique for the identification of recombinant bacteria or yung ating positive transformants. Okay? This technique relies on the activity of what we call the beta-galactosidase, which is an enzyme occurring in bacteria that is capable of metabolizing lactose into glucose and galactose. So, this technique relies on the lac operon, specifically on the lac-Z gene. So, kapag sinabi nating operon, it's composed of many genes. Kumbaga, linked gene siya. Like, one gene cannot work without the other. That's why they are considered as one group, or what we call operon. So to check for positive transformants, we look at the LAC-Z gene. So yung LAC-Z gene encodes for what we call a beta-galactosidase. So tingnan mo, ito siya yung LAC operon natin, yung green, di ba? Part of the LAC operon is the LAC-Z gene. Okay? The LAC-Z gene encodes for beta-galactosidase. Isa lang siya sa many genes present here. Okay? Now, why is this is very important? Beta-galactosidase catalyzes lactose to form glucose and galactose. Ibig sabihin, on normal condition, kapag walang insert dito, galactosidase is able to hydrolyze. Beta-galactosidase is able to catalyze the cleavage of lactose to form galactose and glucose. Okay? And then, paano siya nagiging determinant for the blue-white screening? Okay? Beta-galactose hydrolyzes, ito yung tinatawag natin na X-gal. Okay? Yung X-gal na to, it will be oxidized in the presence of water. So nakikita nyo dito, from this portion right here, nagiging isa na lang siya. Okay? Because of the addition of water or because of the hydrolysis. Okay? So anong mangyayari once this ring is removed from the rest of the X-gal? Kapag nag-break yung band na to, this will turn a blue color. Okay? It will become blue yung ating colonies. Okay? This is true if walang insert sa atin. Ibig sabihin, unsuccessful. Kasi nga, the LAC-Z was able to produce galactosidase which was able to hydrolyze yung X-gal, and X-gal, because na-break yung band na to, was able to impart a blue color sa ating bacterial colony. Okay? This is in the case kung unsuccessful, kung hindi successful. Now, let's try to look kapag merong positive insert. For example, this is our gene of interest. Okay? If merong insert, anong mangyayari? Kasi merong insert, LAC-Z will not be successful in making beta-galactosidase. Beta-galactosidase, in its absence, will not be able to hydrolyze the X-gal. X-gal, without being hydrolyzed, will not be able to impart a blue color. That is why nagiging white siya. So for blue-white selection, a successful transformant may be indicated by a white colony. Okay? I hope we follow. Now, how do we check for positive transformants? The last part will be to further annotate because in here, you can see there's blue and there's white. But just because it's white does not mean it's already successful. Okay, we need to screen more at the molecular level this time. Because later on, for example, if you're going to pick this colony without checking the sequence of this colony, then chances are magiging mali din yung ating downstream analysis or ating downstream applications. Okay? So that's why we need to check at the molecular level. So what we do is we get the colony that has a white and then we subject it to PCR and then we are going to see if it's really true or not. So for example, in here I have 13 white colonies and then I compared it with the blue. So if the band of our candidate whites is the same with that of the blue, it means it's a negative insert. So false positive to siya. This is inconclusive kasi walang band. So maybe mali ako dito in my preparation. This is very weak. That's why I put negative question mark here. Kasi I'm not sure if it's really an insert or not, or maybe nagkaroon na ng contamination. But for the rest, for some, I can be sure na positive yung aking insert. Kasi nga, di ba dito, from top to bottom, mas malaki yung base pair ng mga bands na nasa taas kumpara sa baba. Ibig sabihin, dito, may nadagdag, dito naman, as is siya yung kanyang size. Okay? And this can be further analyzed by looking at the sequence level. So that's why you have to look at the sequence of your white colonies. So this is what I did here. And in here, you can see na some of the white colonies, kahit may positive insert, meron pa ring discrepancy at the molecular level. So, in this case, you have to remove these sequences. Kasi later on, if you're going to study this at the molecular level and then ginamit mo pala yung sequence na to, then your data will be wrong. Kasi nga, meron ngang insert pero yung sequence mali naman from your target. Okay? It's easier said than done, to be honest, but I hope you get what I'm trying to make you appreciate. So once you are able to select your positive transformants with high confidence, then you can proceed by making more of this bacteria. And then when you make many of this bacteria, then you can store it for future use. For example, you're going to run the same experiment years from now, you can still use the same bacteria that harbors your plasmid of interest. Okay? Now, as a review, as for molecular cloning, as we said, there can be many sources of the DNA of interest. Okay? It can be from mRNA turned into cDNA. It can be from point mutations or site-directed mutagenesis. Okay? Or it can be from our environmental samples. So the first step is to put restriction enzymes yung sa ating target here in the block. And then yung ating plasmid naman, we need to digest. We need to digest the plasmid with the same restriction enzyme na ginamit natin dito. Para naman, makonect natin yung black at saka yung green. Okay? After that, we are going to introduce what we call DNA ligase. The DNA ligase, yung function niya is simply to attach yung green part, which is the plasmid, at yung black part, which is yung DNA of interest. So after this one, we now get a recombinant plasmid. Once we get a recombinant plasmid, we put it to our host, in this case, an E. coli. Okay? So kapag nalagay na yung recombinant plasmid natin to the bacteria, this becomes a recombinant bacterium. Okay? And once we do this, the next is to grow this in culture. So we make many copies, many copies of this. And then, we are going to screen for positive transformants. So yun yung sinabi natin na meron siyang ampicillin resistance, meron siyang antibiotic resistance, whether it grows in a media that contains your antibiotic or not. And then second is for the blue-white selection. And if it's white, you proceed at the molecular level by looking at the sequences, if it's really your true sequence, or baka false positive lang siya. And then once it is your true sequence, or if it's the sequence that you want, then you can store the bacteria that harbors your plasmid for later use. And this plasmid can be used in many applications. It can be for pest and resistance control. It can be a gene, for example, that you use to clean oil spills, for example. It can be a gene that you want to help in the dissolution of proteins in blood clots to prevent heart diseases, or it can be human growth hormones or any genes for that matter. It does not have to be a coding gene. Sometimes you can also use it to enhance the expression of a gene, as in the case of regulators, enhancers, and transcription factors. Okay? So if you want to learn more about molecular cloning, or if you have questions, feel free to ask me, or if you want if you want me to include specific topics for UPCAT review, then you may do so. Okay? So thank you very much.